Hello everyone, this is me Akshay Ashok doing PhD at Manipal Institute of Regenerative Medicine. The recent clinical studies have been shown that the people who undergone for organ transplantation results in post-transplant diabetic condition. How pathetic situation that is, right? Yes. And that is now diabetes becoming one of the nightmare for the people who have to go for organ transplantation. What can be done for this kind of situation? Yes, considering the immunological risk would be possible and always maintaining the life measures. Coming to the research part is understanding the key reason that caused the post-transplant diabetic condition. And that is where our area of interest and we found a novel mechanism that could rescue the post-transplant diabetic conditions. Coming to that study, cyclosporin A, which is an immunosuppressant drug, is found to inhibit the calcineurin NF-ATC pathway for organ transplantation. So we decided to look at the importance of the calcineurin NF-ATC pathway for beta cell generation, and we found that when you inhibit the calcineurin NF-ATC pathway, which essentially inhibiting the expression of the key genes that is required for the beta cell generation. Not only that, the insulin cell lines which behaves like a beta cells also showed that upon calcineurin NF-ATC pathway inhibition, there is a significant reduction in insulin expression. So this is clearly indicating that the calcineurin NF-ATC pathway is important for beta cell generation and its inhibition is completely impairing its function and generation. And the further regulation mechanism study of NF-ATC showed that ubiquitin proteasome system is also having a crucial role in beta cell generation by modulating its one of the key component called PSMD2. And we found that PSMD2 is the only candidate that significantly decreases during beta cell generation. But how this calcineurin NF-ATC pathway and ubiquitin proteasomal subunit PSMD2 is interconnecting each other for the beta cell generation and function remains an unknown story. But in our study, we found that the calcineurin NF-ATC pathway is being a savior for beta cell generation and which suppresses the negative regulator of proteasome called PSMD2 during beta cell generation. And the loss of action of PSMD2 is essential for a healthy beta cell differentiation and functioning. So with this, we are concluding that and we'll shed a light on the reason behind the post-transplantation diabetes, where the cyclosporin-mediated inhibition of calcineurin NF-ATC pathway enhancing the proteasomal subunit PSMD2, and that is being the reason behind the post-transplant diabetic condition. So targeting the PSMD2 via small molecule would be a potential approach to rescue the post-transplant diabetic conditions. Thank you everyone.